Thank you for inviting me to give this talk today. I'm excited for it. I haven't actually given a talk like this before because I'm told it's combination science and career path and sort of scientific path, and so it was really fun to kind of go back and put this together. The theme of this is this: what is, or in this case, what are, and the scientific focus I'll have today are oncogenic competency and adaptive plasticity in skin cancer. As a sort of broader strokes here is why do I care about these features and how did I get here? I should say my name is Judson-Torres. I'm an associate professor in dermatology at the Huntsman Cancer Institute and University of Utah. Plan today, I'm going to spend about 20 minutes on professional and scientific path. Sort of lessons learned and sort of scientifically how I arrived at the research vision and concepts that I'm interested in. And then probably about 20 minutes on oncogenic competency. And depending on how much I'm interrupted and how many questions are asked, we'll likely also get to the adaptive plasticity as well to some degree. I really usually dislike Zoom meetings because I feel like I'm just talking to the void. So if you can leave your cameras on and even mics on, since I can't actually see you, I can hear little bits of things here and there. I know you're still there. And please do feel free to interrupt me at any time. Okay, so a little bit of background here. I grew up on a farm in very central Pennsylvania. It is a town that few people have heard of or outside of town called Jersey Shore. Not because it is the Jersey Shore, which is obviously over here, but apparently because at some point some New Jersey folks moved to Pennsylvania and settled on the opposite shore of the Susquehanna River, and so that became Jersey Shore. I grew up there at a time where we really did not, as a family, have a camera, and we didn't all have cameras in our pockets as we do these days, so there are very few photographs. This is one that does exist, that is framed. You can kind of see my reflection taking a picture of it. But this is where I grew up. This was the barn. You can see very much middle of nowhere. And up until recently, I actually didn't think there were any photographs of me as a kid there. But then after my parents passed a few years ago, a friend of theirs reached out and said that they had come to the farm at some point on a winter day with a camera and did send these images, mostly putting these in here for Amanda's sake. But there is in fact proof that I was a kid during the 80s, complete with my awesome NASA astronaut boots. And as far as sort of how this experience formed me, a professional lesson here is I very much knew I did not want to be a farmer for a living. And the scientific lesson was really just a curiosity, right? As you can tell, there wasn't a ton to do here other than to explore, other than to sort of be around a diversity of life. And that's really just what I grew up enjoying. And so when I then wanted to go find something else to do other than farming, I obviously went to university. And what I really wanted was to basically find ways to explore and understand the world. So I went to Wesleyan University, which is in Connecticut. It's a liberal arts university, and I really take full advantage of this. I switched majors 4 times during my time there. I started out as a mathematics major, thinking that sort of these fundamental equations would allow me sort of the best way to explore and understand the world. And I found pretty quickly there that although I could understand and reproduce mathematics quite well, I found that I didn't really think I was advanced enough to be able to discover anything new, and that turned out to be something I really wanted to do. And so I switched majors into philosophy and enjoyed this for a good minute or so, where it's a relatively low barrier field for trying to sort of develop and hone one's ways of thinking up of new thoughts and new ways of thinking about the world. But in this case, it really bothered me that in the end, it was essentially just argument. There was really no way to prove or disprove one concept over another, just sort of different ways of wording it. So then I switched into economics. It seemed to me that economics was a combination of philosophy and mathematics, and I could maybe come up with new thought and then find evidence in the world that could support or not support the ideas. And I really did like economics. I ended up minoring in it. But one thing that really bothered me here is that what I would come to realize is that you couldn't really have, or at least at the time, although I think this is changing, a perfect experiment, right? You could look for data sets that existed in the world to see if they supported your ideas, but you really couldn't do what is ultimately a controlled experiment. And that's when I found myself drawn to molecular biology and biochemistry. So growing up, I really didn't have much of a science education to speak of. My first exposure to molecular biology came my sophomore year of college, and it really just really spoke to me, and specifically a fascination with a particular molecule called the hepatitis delta virus, which I'm going to talk about here in a minute. But sort of the professional lesson here is, through all this jumping around of majors, I just kind of worked my way down to realizing I had a very sort of innate fascination for an inclination toward both a reductionist approach and a hypothesis-driven approach to exploring the world around me, and that was incredibly valuable. Now, with this, I wanted to ask questions, and the one question I really wanted to ask was a simple one and not a particularly creative one, which is what is life, or more specifically, how is life? And I wanted to do this with a reductionist perspective. And so my research in college was, as I mentioned, really focused on a fascination with my first love here, this little guy, the hepatitis delta virus. So I imagine most people are familiar with hepatitis B, that is shown here. And this is, we just call it a fairly classic virus. It's got a genome that encodes for some proteins, but not all the proteins that it needs. It infects cells, and hijacks cell machinery in order to be able to reproduce everything it needs. The hepatitis delta virus is even more simple. It is one of the reasons I was drawn to it. It's the smallest genome to infect humans, right? So it's only about 1.7 nucleotides long. And it does not at all produce everything it needs. It's really actually more of a parasite for hepatitis B. What this diagram is showing you is all of these are the AG here, these different antigens. These are different proteins produced by hepatitis B to make its envelope so it can infect other cells. And hepatitis D just comes along for the ride and steals these from hepatitis B. It's a really interesting relationship. In fact, if you get a high enough titer of hepatitis D, it starts to reduce the amount of hepatitis B in your system in a sort of classic predator-prey relationship. And one of the things that really, really started to fascinate me about hepatitis D, and this is an image I took yesterday out of my undergrad thesis, is that you have this tiny little genome, 1.7 nucleotides, and it's RNA. And it's a circular RNA that can fold into a variety of different structures. And based on those structures, it does different things, right? It can fold into structure 1, which is required to basically yoink and steal away these various proteins from hepatitis B. It folds into a different structure in order to replicate. And as part of that replication process, it even folds into a variety of structures with enzymatic activities, right? It does not encode many proteins, but the RNA itself can fold up and essentially perform the same function that we'd normally attribute to a protein. It's called a ribozyme. It's the first ribozyme discovered. And so, at the end of the day, nothing really tangible came out of this. My research was never published. There's nothing I could put on my CV. But what really struck me was a career-long fascination for RNA. So I'm going to show you the variety of different types of research I've been involved in over the years. And there is a variety, but they all have this sort of central idea that really came from my first fascination with hepatitis delta virus. And that's really that the scientific lesson that RNA does everything. So I stumbled across this comic strip a few years ago and absolutely loved it because essentially this little guy here in the 2000s, I'm pretty sure I said exactly this. This could have been me, right? So if your education was anything like mine, we learned about the central dogma of biology that started in the 1960s, where the idea is that DNA is this fundamental molecule of life. RNA is then a messenger to make proteins from that DNA. In the 80s, perhaps started to note that RNA does some other things, and yeah, okay, interesting. But really, you can see where my training started 2003 in the 2000s. This is really when it became obvious that there are a ton of different types of RNA, and they can do all the things that we initially thought that only DNA could do. They could do the things we initially thought only proteins could do. And that really, a lot of my work also contributed to where we got to ourselves in the 2020s, which is this RNA world idea that really life is just a seething mass of RNA that sometimes uses DNA to take notes, and the proteins are just the errand runners. So this is essentially my biologic worldview that has been really sort of fundamental to all the different projects that I've done during my career. So after all that major switching, I was graduating knowing I loved biology, but clearly I'd been infatuated before. Having switched majors many times, I wanted some additional experience to know whether I really would, my love for research would stick. I was also really interested in sort of the ecosystem by which research is conducted. It has always seemed non-obvious to me that this would be something that is sort of so well supported and funded as much as it is. And so I figured the place to go sort of learn this ecosystem would be the National Institutes of Health. So I spent 2 years there. I wanted to reduce my question of what slash how is life even more. And so I targeted the lab of Henry Levin, who studies retrotransposons. And again, I won't spend much time on this, but it's another fascinating type of life, which is essentially just a strand of RNA that can form a ribonucleoprotein protein complex. It gets integrated into mammalian genomes, including our own genomes. Our genomes are filled with these retrotransposons. And there they just get propagated as DNA. And at some point in various stages of evolution and or development, they can get reactivated and form another copy, right? So you're really down to just this very basic idea of a molecule that can replicate in some way, shape, or form, being a fundamental bit of life. Now, this was also a hot mess. I spent 2 years there. Again, nothing to speak for on my CV. I learned a lot of lessons here. The educational value of failure. Things like controls are important, making your own reagents is important, notebooks are extremely important. Lessons I'm really glad that I learned before I got to grad school. But unfortunately, the cost of it was not a ton of published productivity from my years at the NIH. And then scientifically, as you know, my conclusion from my time there and speaking with a lot of brilliant folks who are there is that if you want to continue to try to reduce this question of what is life, replication is really the final most irreducible property of life. And that I found to be interesting, but not particularly satisfying, a little underwhelming. It's a bit of a tautology, right? So you're kind of defining life by the fact that you see it in the first place. And so when I was looking for where to go to grad school, I kind of shifted this sort of driving why of why am I a researcher, and instead wanted to ask the question, why and how is life interesting, right? So, okay, so if it's just replication, fine, but how do we get such complex organisms then? And this has really driven my research since. And so what I'm showing you here is a human blastocyst. So this is approximately what we all looked at like at day 5 of life. And if you've not had training in developmental biology, the main thing I want to focus on is this little thing here. Oh, by the way, can you all see my cursor? Should have asked that earlier.
Yes.
Yeah.
Yep.
Great, thank you. Okay, so this little mass, this is called the inner cell mass. And this little blob of cells are the cells that will go on to form every cell type in every tissue of the adult organism. And to me, this entire process is fascinating, right? So this is where you would derive an embryonic stem cell from. And this is where if you sort of drop in an embryonic stem cell from another organism, you can get a chimera of the adult. You can form clones, that whole bit. And I just became very interested in wanting to understand this sort of dichotomy between, to have an adult complex multicellular organism, you need to have cells that are, or these life forms, right, that are working together on this incredibly complex and fine level. But at the same time, they also have to be really adaptive, right? You have to be able to respond to things in your environment and have the cells change what they do without losing control of those cells and them forming what ultimately would be a cancer or just a disorganized organism. And so this is something that I really wanted to look into. And so I decided to pursue my PhD in stem cell and developmental biology at the University of California, San Francisco. This is under the umbrella of the biomedical sciences program. And this is an incredible time in my life. 3 sort of main things happen professionally here. I'm going to talk about a little bit of each of these. This is where I really developed an even more keen awareness of really the surrounding research ecosystem that we all conduct our research in, and the influence that, an often unseen or unappreciated influence that has on our scientific discoveries. It's really the first time I made any, much less a major contribution to research in human knowledge, so that was pretty fun. And by the end of it, I was also extremely jaded with basically the whole academic scientific enterprise. And so I'm going to talk about each of these in order pretty briefly here. So the first is the ecosystem, right? So I just speak as, I think a lot of the ecosystem around us can be taken for granted. It has not always existed in its form. And by ecosystem, I mean, where does our funding come from? How is that funding distributed? What are metrics for success, right? How are things published, the review system, so on and so forth, right? Each one of these things was created, and it didn't have to be created the way it was, and they have evolved, and they didn't necessarily have to evolve the way that they did. And I can talk more about this if anyone's interested. Again, always feel free to interrupt. Happy to stop on any of these issues. But what I essentially felt like I noted as a PhD student is that there were big problems with these systems, right? A lack of communication between the ivory tower and the public, a sort of type of science and type of story that were really elevated by particular journals, and metrics of success that were basically gameable and not necessarily metrics of contribution to human health and knowledge. So I spent a lot of time on the Hill. I spent a lot of time trying to pick apart the publication system. I had startups. I spent a lot of real time trying to understand and solve these problems. By real time, I'd say about 20 to 30% of my effort as a graduate student the entire time was spent on these sorts of projects. There were a few tiny wins, but as a whole, I really achieved nothing of lasting value here, other than, again, learning that well-earned failure can be an invigorating process that teaches for the next time around, that there's intrinsic value trying to solve big problems, even those that might appear unsolvable, and particularly a skill for how to take seemingly unsolvable problems and break them down into smaller. And I'm really spending time on this because, at your stages in your careers, I can tell you with certainty that my efforts in this area, which had nothing to do with my research, did as much, if not more, for accelerating my career than anything else. And the reason for that is that in attempting to understand the system and disrupt the system and probe the system, I really got on the radar in a positive way with folks at a lot of other institutes other than just where I was at UCSF, right? So it took me all around the Bay Area, PLOS, AAAS, National Academy of Sciences. There were people at each of these programs who essentially knew me in a positive way as a consequence of these efforts. And so, one thing to keep in mind as you're pursuing your own careers is, you need to have a baseline of excellent research and proving yourself as an excellent scientist. But there's going to be a lot of folks in that category, right? And finding something, preferably a passion, that kind of gives you as a signal above the noise, so to speak, really came back and ultimately saved my career. And I'll talk about that in a slide or two. But meanwhile, scientifically, what was I doing? Again, I was trying to ask this question about how a stem cell becomes and can sort of solidify as all these different cell types in the adult system. And as I've mentioned, I already felt like I knew the answer to this. I just needed to prove it. And I needed a system to do that, right? And having a system or an assay that is reliable is probably the most important part of pursuing your PhD. And I was lucky in that my system was developed essentially the same month I was starting grad school. So I was going to say show of hands, but let's do show of voices. Do any of the trainees who are here know who this person is?
Nope, no idea.
Yeah, so one person does not. No, I appreciate it. So this is Shinya Yamanaka. He's a Nobel laureate, and this was a seminal paper published in Cell. I'll point out the sort of lack of recognition here is more evidence that if what you are seeking in life is fame, then science is not the place to be. Because as scientists, I think we rarely even recognize our most famous scientists. But Shinya Yamanaka made the seminal discovery in 2006. And the title really tells you what it is, right? He's able to take fibroblasts, so skin cells, and turn them back into embryonic stem cells in a very controlled and defined way. And this was absolutely earth-shattering to the field at the time. I realized that you were probably all about the age of Super Boot Sarab here at the time this came out, so this is all old news to you. But at the time, this idea that any cell in the human body could be turned back into an embryonic stem cell was, I mean, just basically turned the field upside down. And so I immediately latched onto this. I was reproducing these data within a month or something of it being published. And the assay roughly looks like this. On the left, MEFs are mouse embryonic fibroblasts. The Yamanaga factors are 4 transcription factors that you introduce into the cells artificially. And we also have a transgene in there that will turn green if the cells become embryonic stem cells or iPS cells. And that's what happens, right? You add the factors and you get these beautiful green colonies. You can then take those cells and prove that they're embryonic stem cells because you can clone an entire mouse. So this is my first cloned mouse. We know that it's cloned because it's blue. I put a cassette into the genome of those iPS cells that allows me to stain and see what tissue in this mouse came from the cells that I put in, and it's all the tissues. Um, Now, what I became really interested in, though, is that although this is true and an incredible discovery, the truth is it almost barely never happens. Even if every single cell gets a healthy dose of these Yamanaka factors, only about 0.1% of them actually turn into an embryonic stem cell. Another 0.25%, sorry, an animation isn't working here, but that arrow is pointing toward essentially something else that's a colony but not green, something that might be like a transformer cancer cell. You get a lot of apoptosis and senescence, but the vast majority of these cells just do nothing, right? They stay as mouse embryonic fibroblasts. And so I became really interested in asking what is permitting these cells to basically stay so robust in their cell identity in the face of these factors. And so these 2 images are ones that essentially changed the trajectory of my career. What you're looking at here, what I've outlined, is a full well of I think it was a 24 well plate, if I recall. And what I was doing as a grad student was basically sitting in a dark room and scanning through the entirety of these wells and counting the number of green spots, and to see if as I can tell you that all the black space you see here, those are fibroblasts, and so you can see there's only a small handful that have turned green. And it was actually during my rotation where I tested my first hypothesis. And the hypothesis was that if I took just regulatory RNAs from ES cells and transiently put them into these fibroblasts, that it would make this whole thing work better. And this is just one of those aha moments that I'll never forget. Is I, got in the morning, looked in the microscope, and this is what I saw instead. This was earth-shattering to me. Basically, over the course of my graduate work, we published 4 major papers on this. I'm not going to get into all the nuances. A lot of it was digging into mechanism. But to me, the real sort of conceptual thing here is that we showed that you could take transient addition of blastocyst or ES regulatory RNAs and fully change a skin cell into an ES cell. Or we could use transient inhibition of regulatory RNAs in a skin cell and also turn those into an ES cell. And so what that tells us is that every cell in the organism, it really holds this potential of really all cellular behaviors within the full organism. And it's restrained by transcriptional barriers, such that if you just, transient loss of each barrier can be sufficient to unlock an alternative cellular program. And what struck me at the time is how much this contrasted to this idea of gain of function when we think about the sort of prevalent model at the time of how cancer evolves, which is this idea that there's a series of genetic mutations that each confer a different behavior onto these cells and that is selected for. And so I felt that this was a very important and interesting concept to begin to study in cancer. I was not a cancer biologist at the time. But I really didn't think I would ever get there because at this point in my career, I was absolutely ready to throw in the towel on this entire endeavor. I'm not really going to get into all the details on this. Happy to talk about it later, but I think it is important to acknowledge these things, especially to trainees early in their career. That there are definitely very frustrating things about our system and our choosing career paths. I was in the UC system. You're probably all aware of these strikes that occurred a few years ago. It took a long time for things to bubble up to this level of strike. The treatment of trainees and how they were functioning in the system was very difficult. I also felt that the field as a whole was quite frustrating. There was a particular conference I went to where, this is in sort of the stem cell regenerative medicine field, and there's always sort of touting of the promise of stem cell therapy, and they bring in patients who would say, "Thank you for all that you do. You're really doing things that are going to help us." Patients would leave the room, and then you would just have scientists kind of, I don't know, yelling at each other, right? It was really a rather toxic environment at the time in terms of the cutthroat competition, and I found that really disheartening. Sort of the maybe funniest version of this was again this blue mouse, my first cloned mouse. My mom actually printed this off and put it on the wall because she thought it might be the closest to a grandchild she'd ever get. And my dad asked, what exactly are you doing over there in the big city? I'm like, oh, cloning mice, right? Again, I'm a farm boy. We always have mouse problems. And he's like, why? It's like, well, it's a really good point. I'm not sure I could tell you why. And so I was just generally disheartened with both the system as well as even really what I found to be incredible scientific discoveries, how they would ever translate into something that could be useful. And so I decided to head out. I graduated, got my PhD, and I took a job as a geneticist at the direct-to-consumer genomics company 23andMe down in Silicon Valley. And my job had two main components. Part one was to be a geneticist, right? So for those who are unfamiliar with GWAS, genome-wide association studies, these are this sort of idea that the genome is going to have an incredible amount of information that if we could unlock it would tell us useful information about either our traits or disease or likelihood of responding to a therapeutic and a variety of things like that. And GWAS studies basically take a phenotype and then look across the genome for alterations that are associated with that phenotype. And my job was to take all of the best GWAS studies published in the best journals in academia, redo all the stats, see if we could reproduce it, and most of the time we couldn't, right? So that itself was a little disheartening. But on the flip side, sometimes it could be reproduced, right? Sometimes there were associations that were reproduced by multiple independent academic labs, as well as the data set we had at 23andMe. And in those cases, I had a part two to my job, which was then to try to report this to a user of 23andMe. And this turned out to be incredibly challenging. I essentially had to figure out or help figure out how to explain odds ratios and concepts like lifetime incidence to a layperson. And we were charged of like this has to be explained in 20 seconds and less, and they have to click on something else at the end of it. Not a particularly easy task, right? And you end up with little figures like this one. Where you say, okay, if there's 100 people exactly like you, and then and one of these people will get this disease or have this trait, and then there's 100 people exactly like you, except they have this one SNP that we found or this one genetic alteration that we found. Three of those 100 people will have that trait or that phenotype. In a variety of A/B testing of speaking with users, I'm trying to get them to understand why this was useful, important information that they might care about. It honestly really felt to me like I was becoming surprised at sort of the inability to explain why, on an individual basis, this was particularly useful information. And it really surprised me that given the immensity of information that we know is encoded by the genome, how little predictive power it has. And I do think we've seen this play out. If 23andMe has recently gone bankrupt and they were really had all the resources and all the genomes needed to test this hypothesis that if we look hard enough at the genome, we can produce information that will be predictive and importantly useful, and due to useful also valuable. And it really had not panned out particularly well. And so I found myself like this cow, realizing that I was really looking at the other side of the fence and thinking the grass was greener, and realizing that I really, really missed academia. And the main thing I missed was this, right? It's the academic part of it. I had been so focused on problems with the ecosystem of the research part, I'd really failed to realize that I'd been an academic literally my entire life, like from kindergarten to age 30 or something. And how much I just didn't realize how special the relationship was, I guess I should say, between a mentor and a mentee. And I'm not going to list through these, but you can read them real fast, right? There is this idea of there's a big difference between a mentor and a manager. And it's not that this is an exclusive, academia as mentors, industry as managers. You definitely have academic labs that you have are led more in manager style, and you've got industry labs where people take on strong mentorship roles. But generally speaking, in most academic situations, you are paid to do the job of a mentor, and in most industry positions, you are paid to do the job of a manager. And these differences are not trivial, right? They to me made a huge difference in my uh, just enjoyment of sort of day-to-day life at work. And so that made me want to go back to academia, which is not necessarily the easiest thing to do. And that's where I mentioned this earlier, all this work that I had done on trying to find ways to sort of challenge, study, and disrupt the academic publishing system that really put me on the radar of some important folks at some major institutions really turned out to be critically important. It was due to those relationships that I was given the opportunity to essentially treat my time at 23andMe like a postdoc and then start up a lab at UCSF as a PI. Again, I do think it required that I had a solid science, publications and scientific background. But if that had been the only thing I had, I don't think I ever would have had this opportunity. So now we're almost 11 years in. I had my lab at UCSF for about 4.5 years. I've been at the Huntsman Cancer Institute ever since. People ask me sort of the big reasons for the changes, and I won't get into most of them, but I did mention how I'm really interested in the ecosystem of how science is conducted, and UCSF is a wonderful, also very well established place where it was really difficult to make changes. And Huntsman really spoke to me as one of the newest cancer institutes in the States, and a place where we're still building an environment, and leadership here wanted to build a new type of environment to conduct research. And so being able to come here and help build from the ground up a community of solutions, I found really enticing. Stepping back in from industry, I was able to set up my lab with the values that I wanted to have, which is a people forward lab, so mentoring via research. And essentially, this is the most rewarding part of my job, right? So here's this collection of current and past trainees. There are others sort of taking a snapshot here, but really helping to have trainees, whether they are undergraduate, graduate, postdoc, clinical fellows, sort of reach their full potential is easily the best part of my job. And I just threw this in here to hopefully embarrass Amanda a little bit. This is the party after she just had her first author publication, and as doing so, got to sign that hat that she's wearing, which I have everybody sign. I also got to choose what I got to research, right? So I was trained in developmental and stem cell biology, but I really felt some of the concepts I discovered would be useful for cancer. And I decided I did want to explore this idea of sort of the legitimacy of this genetic evolution of cancer model. And I was going to do so, I decided, by studying melanoma. And so what I'm going to give you now took a little bit longer than I expected, but it'll be a pretty rapid summary of the last 10 years of research in my lab with a focus on oncogenic competence and adaptive plasticity. And again, if I don't get through it all, that's fine. You're welcome to interrupt me with questions. Before I get into that, I have to give a thank you to all of our funders. That's in the bottom right, and on the left, all the collaborators and lab members and alumni who participated. They're the ones who actually did this work. So why focus on melanoma to begin with? It's not the most common disease. We have about 7,000 deaths a year in the United States. It is among the more common diseases in young adults, which I think is a particularly tragic age to develop a lethal cancer. So that's one good reason. A second is that the incidence has been rising year after year for quite some time. And that's particularly true in Utah, where we've got a 37% higher rate of melanoma. We're the highest in the country, unfortunately, for the amount of melanoma. So those are all great reasons. But really, the scientific reason I want to study melanoma is that it's a pigmented cancer of the skin, and it has different defined stages or phenotypes, leading from what I have here on the left of the epidermal melanocyte, the pigment-producing cell of the skin, through a hyperproliferation stage. A growth arrested stage is a nevus, also called a mole. Escape from that growth arrest into another hyperproliferative but non-malignant stage called melanoma in situ, invasive melanoma, and then a series of events to lead to widespread metastasis. And because this is on the skin and these stages are well defined, it theoretically would allow me to have access to fresh tissue from each stage and really begin to try to pick apart the relative contributions of the genome versus the transcriptome to each of these evolution stages. So it took some time to set up the pipelines for this. We have a variety of different ways of doing it, and I'll move through this pretty quickly due to time. But essentially, we can find what we call transition cases, where you have multiple different stages of the same disease in one H&E section. We can microdissect that out. We can get fresh tissue of healthy skin or diseased skin, frozen tissue from different sources. And we've developed protocols for pulling out the different types of cells and studying them individually on a single cell level. And so using those tools, I started by assuming what I guess I call the null hypothesis here, that cancer is just a genetic disease, right? So that you're going to have a mutation associated with each phenotype that is reproducible, measurable, and responsible for that phenotype. Right off the bat, I sort of knew that this probably wasn't going to pan out. So melanoma is a bit of an umbrella term for a variety of different presentations of pigmented skin cancers, all of which are theoretically are arising from that melanocyte. And the genetics of these have been looked at for some time of the advanced disease. And having been around when some of these studies were being done, I know that the assumption was that there was going to be a different set of genetic drivers or secondary mutations associated with each of these different types. And that has generally not been true, right? What I've highlighted in red here are those cases where you do have a bit more of a trend, right? So non-chronically sun-damaged melanomas are 40 to 60% driven by BRAF. Desmoplastic melanoma is 50 to 70% driven by NF1 loss, things like that. But you can also see that there's a distribution across the board such that if you were to try to ask whether you can predict the phenotype by the genotype or if you can predict the genotype from the phenotype, even the best case scenarios, we're looking at pretty comically low sensitivities and specificities, right? So there really is not a strong association here. Now, there's just enough of association that if you come into this assuming that the genetics are really the cause of this disease, you can view it as this is a glass is half full. Given my background, I was viewing it the other direction. Another feature of melanoma, it has one of the most highly mutated genomes of any human cancer. And so this has been assumed to be a feature of the disease, and again, something that's causal, right? If you have a highly mutated genome, that'd be a good reason to be a cancer. However, we noted pretty early on that these studies came from comparing the tumor to germline, either through blood or through human skin of the donor. But where each one of those cells, if they did have mutations, they're not clonal, right? They're going to have a different contribution to the sort of bulk sequencing you're looking at, in which case it's not really the right denominator. And so we decided that we would sort out melanocytes of healthy appearing human skin and look at the mutation burden in each of those genomes. And what we found is by and large, by the time we're about 30 or 35, the mutation burden of healthy melanocytes in skin is already just as high as melanoma. And so what that tells you is, yes, there's a high mutation burden, but it's not actually a feature of the disease. It's a feature of the cell type. So that's sort of random UV-induced mutations. But what about known driver mutations? To do that, we microdissected out each of these different stages that we talked about and conducted genetics. And again, the hypothesis would be that each of these different stages would be associated with a new mutation that would be reproducible. That's not what we found, right? This is a very well-established gene, BRAF V600E. It drives about half of melanomas and all of moles. And we found that this was really the sole mutation through most of these early stage phenotypes, which shows you that you can have a single mutation with multiple different phenotypes. And then with the switch to invasive melanoma, 60% of those, that's a pretty good signal, have loss of the CDK2 tumor suppressor. And that really seems to be the last recurrent genetic alteration through the rest of progression to metastasis, with a full about 25+% of these unexplained by genetic alterations. Moreover, this is more recent data that Amanda was also involved with that's currently under review. More recently, we have begun to ask, what about this assumption here that the BRAF is occurring and causing melanocytes to then hyperproliferate and become nevi? Do we really know that, right? Have we been conducting the right experiments? And so we decided we'd start to look at skin that had no clinical evidence of a melanocytic lesion stained for BRAF V600E, and we began to find BRAF V600E positive melanocytes within that skin. And so we started a much larger study where we took patients who had previously had a melanoma, but the melanoma had been excised and there had been no recurrence for 10 years. And we took skin adjacent to that scar, proximal to that scar, or distant from that scar, and used technologies to look for a number of BRAF V600E positive melanocytes. And again, found that in about 75% of the skin we looked at, whether it was scar adjacent or further away, also contained BRAF V600E positive melanocytes. And so essentially, this arrow gets moved back even further, right? That our skin has fields of these melanocytes that contain oncogenes that are otherwise just fine. And so what that tells us, right, is that the genotype-phenotype relationship, at least in melanoma development, is bidirectionally non-deterministic, and that the features originally measured in advanced disease really are not unique to that advanced disease. And one might argue we already knew this, right? So BRAF V600E had been discovered a number of years ago, and strong inhibitors against the mutant kinase have been developed and used clinically for some time. And again, was around when this was first developed, and I just remember how excited everybody was because you could take a cancer that did not really have any good therapeutic options, treat the patient and have it completely clear. And then also around when, sort of the other ball hit the floor, which is that everything generally comes back, right? This is usually about an 8-month response or so before uniformly tumors are going back in all the exact same locations. And so, the simple truth of it is, for all of the melanoma genomes that we've sequenced over the years, of which there have been hundreds, if not thousands, we haven't discovered another actionable alteration, or quote unquote driver mutation, that has really led to any sort of sustained response. And we have a wonderful control here, which is immunotherapy, right? During the same time frame, not looking at the genetics, but instead targeting the patient's immune system, has led to the development of therapeutics that have led to the first truly durable responses and cures of this disease that we've ever seen. And so I think that really leaves a question, are we done here, right? Have we exhausted the utility of looking at the melanoma genome as a way of trying to understand and treat this disease? And this is what really sort of led me to this question of oncogenic competency. I'm forever an optimist, so I want that answer to be no, we're not done here. And so I'm going to lead you through just a series of experiments here that really sort of changed the way that we began approaching things. So this picture here in the upper left, these are primary human melanocytes taken directly from healthy human skin, so P0. And I know it's a little bit hard to see, but you can clearly see all these dark ones. That's the pigment. There are other melanocytes here that you might be able to see with squinting that they're hard to see because they're not pigmented. And the reason for that is we have found that we have different types of epidermal melanocytes in human skin. We can then take these cells and induce BRAF V600E using a DOX-inducible construct. And we can also found media conditions that allow us to switch the transcriptomic states back and forth between these pigmented and non-pigmented melanocytes. And what we find is if the cells are in the pigmented state and we introduce the oncogene, it's not an oncogene, it drives growth arrest. Whereas if the cells are in the non-pigmented state, the oncogene drives hyperproliferation. And we can switch back and forth. So in this experiment, all the cells are pigmented. We introduce the oncogene, none of them are growing. And then in half the wells, we switch to the other media, and all of a sudden these cells start proliferating. When we look at the transcriptome, right, DOX genes, these are the genes that are changed as a consequence of just introducing the oncogene. Genes change, but this is pretty much dwarfed by the change in the transcriptome based upon sort of these tweaks to the environment. And so that really led to this concept of oncogenic competency, which is essentially the inherent or acquired ability of a cell to respond to an oncogenic stimuli in a way that promotes malignant transformation. And the reason I think this is so exciting, right, is that our data would certainly suggest in the context of melanoma that it's not the acquisition of the oncogenic mutation that's rate limiting, but it's actually the sort of frequency with which that is coupled to cells that are oncogenically competent. And because this is a transcriptomic change, not a genetic change, it might actually be reversible. So here's just one example of it. These are genetically engineered mice. They're engineered to be hairless, but they still have a competent immune system. And they form each of these as an individual melanoma that progresses toward metastasis over time. And so we looked at the mechanisms that controlled oncogenic competency, and we developed a topical that would deliver a small molecule that blocks one of the key mechanisms of oncogenic competency in these mice. And then we looked at in the same mice at the development of melanomas in a treated region versus the rest of the mouse that is untreated. And what we found, so here in the controls, both regions, your melanomas grow. And then in the experimental mice, the regions we're not treating with that topical, the melanomas grow. But we've stopped growth in those where we're treating it. And so this is very new data, currently under review, but it gives me a lot of hope, right, that we can use a topical application to prevent transformation, even after these oncogenic alterations have been acquired. So conclusions and current directions from this first bit is that melanoma has a very high mutation burden, but these mutations are neither exclusive to the disease, nor have they been particularly informative. And they have informed us on trends that serve to guide biological questions, but these relationships between genotype and phenotype really are not that strong. On the flip side, we're living through an incredible time for cancer research, right? I think one of the reasons we've been so focused on genetics in the past is it was among the easier things to measure due to high signal to noise ratio and the fact that it's irreversible. But technology has overcome this now, right? We can measure a lot in cells now, all sorts of different molecules with single cell resolution, temporally, and with rigor. And so I think there's just a world of opportunities in this direction. So I've told you a bit about oncogenic competency and how we're moving forward with asking whether we can target subclinical primed melanocytes with these topicals. And in these last few minutes, I'll probably only have time to sort of introduce you to the concept of adaptive plasticity, and then I'll kind of skip through some data. And so sort of parallel to all of this, I was asking this question whether the concept of Darwinian evolution as a model of tumor progression really was applicable to melanoma. And just to introduce you to this concept for those who might not be in this field, there are different types of models and different types of proven forms of evolution, right? As cancer biologists, we often think of genetic evolution. This is what I've been talking about, right? Where you've got a change in the genome, and that is associated with a change in cell behavior or phenotype. But there's also a different type of evolution called adaptive plasticity, where there's no change to the genome, but the phenotype is changing nonetheless. And what's really interesting about these types of evolution when you look at them in nature is genetic evolution. This is what changes species, right? So, and it's associated with a number of different traits. You often have a high mutation rate. It's irreversible. It's generally better for the generation of a stable new trait and a stable new trait in a uniform environment. Whereas adaptive plasticity is often used when they are rapidly fluctuating environments. It's reversible and it's associated with the low mutation rate. And so off the cuff, when considering everything that a cancer cell must adapt to when metastasizing and becoming resistant to a therapy, adaptive plasticity, at least to me, hypothesis, feels like perhaps a more sort of a priori reasonable model to go after. And so one way of looking at this is to ask about the diversity of the transcriptome at each of these different stages of progression. So we've done this with spatial RNA sequencing, and each of these is a different patient, but the net effect here, this UMAP, which again, it's going to plot the closer cells are together, the more similar their transcriptomes are, is that when we're looking at stages before invasion, regardless of what patient you pull from, the transcriptome of the nevi and even dysplastic nevi are pretty similar. And then when you move into this invasive melanoma bit, you see this sort of diversification of transcriptomes. And I want to point out this isn't in any given direction, right? There's actually not much similar between these different cancers. What's similar is that they are all different from each other, where that is not true when we're just looking at the early stages. And so we've done this now with just healthy melanocytes in human skin. No matter who we pull from, the transcriptomes generally look the same. Also true of nevi. And that similarity is just completely lost on this transition to invasion. And so we're interested in asking what's the mechanism here? How do you go from a cell that has a tight control over its transcriptome to cells that do not have a tight control over their transcriptome? And this actually looks like genetic evolution, which is pretty exciting. So I mentioned before that in 60% of cases, the switch from melanoma in situ, its evolution to invasive melanoma is due to loss of the CDKN2A tumor suppressor. You can kind of see that here in this section. Everything you see on top here is melanoma in situ. Down here on the bottom is invasive melanoma. And then in brown is P16, which is the gene encoded for by CDKN2A. And you can see that it's clearly here expressed in MIS. It's lost in invasive melanoma. Now, one of the reasons this hadn't been really sort of considered before is that we as a field really think we know what CDKN2A does. It expresses for a very well characterized protein called P16, which inhibits CDK4/6, which just blocks cell cycle progression. But one of the interesting things that's been sort of known for some time is, but I think hardly ever acknowledged, is that there really isn't much of a relationship between CDKN2A loss and proliferation of melanoma. So in this case, Ki67 is a marker of proliferation. We're staining sections like this, and we find that in MIS, for example, these are always highly proliferative, but they also still express P16. So I'll move very rapidly here. We essentially tested this by using CRISPR-Cas9 to knock out CDKN2A, and we find that we do not see effects on proliferation, except in very particular environmental situations. But instead, we tested this concept. Let me just skip through some of this. Of is there an effect on adaptive plasticity? So the ability for a cell to adapt to new situations. So for example, here we have a reporter for one phenotype. This is flow data. We're uniformly in this, we'll call it this M cherry high state. We then switch to a different media, and for the cells to survive, they then need to adapt into this low state. Some cell lines uniformly adapt. Other lines, only half the population changes. And when we look at their genome, the only difference between these two types of lines is loss of CDKN2A versus not. We can also then take these cells and ask what's the molecular difference between the cells that did adapt and those that don't. And again, CDKN2A is an answer in both the RNA and the protein level. And again, using CRISPR, we can introduce this phenotype, we can rescue the phenotype. And the sort of cool kicker here is that this is not a gene loss associated with one phenotype. It's a gene loss associated with the ability to rapidly acquire any phenotype given selective pressure. And that's what we've tested here. We've gone around the wheel of hallmarks of cancer and found that no matter what we challenge these cells with, in the controlling for proliferation, in the absence of CDKN2A, if they do not have that gene, they're better at adapting to the new environment. And then here we show that that is also true of metastasis, that if the cells can express P16, they do not metastasize. If they can't, they do, and we can both rescue and replicate that experiment. So that is adaptive plasticity in a nutshell, and why I think it's really interesting. Generally speaking, when treating cancers, resistance is always a problem. In this particular case, we're using this knowledge to develop therapeutics that wouldn't in of themselves do anything to the cancer, but they would prevent these cells from being able to adapt any further. And so our hope is that if we couple that with current therapies, that it will increase the efficacy of really any targeted therapy. And then sort of main take home, I would argue from my just reflecting on sort of career progression, it was just advice is that if something doesn't add up to you as a scientist, you should question it. And this is really true if you see that no one else in the room is. In my experience, that's led to a lot of mistakes, but also discoveries. But either way, it was the willingness to think about and question things in my ecosystem or my science that others weren't, that it really allowed me to sort of continue on this career track, which I love. Okay, I think I just made it under the hour, so I'm going to exit out now. And one more thank you to all of you. Happy to take any questions.
Thank you. I didn't understand a lot of science, but I surely enjoyed your the talk on the career path and all those little images. You know, my son would have actually enjoyed it too. But I actually, you know what? It's interesting that you mentioned. One of my questions was, you know, what you learned along the way, and you clearly demonstrated that, you know, even though you had failures, you actually learned so much from those failures, right? And I'm glad that you reflected on that, because my question was, you know, as you were going through your career path, and you know, what would be your advice? And you just gave it to the trainees. You know, it's okay to fail,
Yeah.
right? Ask question of what and why.
You know,
That's my biggest takeaway from your presentation.
I have a 5 and 7-year-old daughter, and they ask me what I do, and I tell them I make mistakes and I'm wrong for a living, right? My argument is that if the science you're doing, the research you're doing is truly innovative, and you do it really well, like there's no, you know, no technical failure. If you're not wrong 80% of the time, then it's probably not all that innovative, right? And so if you're really trying to push the envelope, that's what we're paid to do, right? It's to come up with hypotheses and test them well and be prepared for them to be wrong. Unfortunately, they've now interpreted that I'm wrong about everything all the time, and so I never get my
that's where you could have. That's where we should always. I'm always right as a parent. Let me just be. Let me make it clear.
Yeah. Parenting is not a science. Science is often wrong. Parenting is not. Yeah, yeah. But no, thank you. I appreciate that. Yeah. It's again, the way I'll word it to my trainees, Amanda can attest to this, right? That whenever you are in a career transition, the more people who are thinking about you and batting for you, the more likely you're going to find that golden opportunity. And you only get those people by getting out there, right? And there are egos out there that if you challenge what they think is true, they'll be angry. Okay, you know, fine, they're out there. But I'd say most of us don't fall in that category, right? And if you're really coming with an idea that challenges anyone's way of thinking, they know who you are now, right? And there's a lot of respect for that sort of level of thought, and that can come back and help in a lot of different ways.
Yeah. Guys, you guys have any questions? No. Okay. Well, thank you so much. We appreciate your time. I truly enjoyed it. You know, as I said, it's amazing how you went from one to another. You really went through all different fields and different career path that you tried to pursue, and so we truly appreciate that. And we appreciate you supporting Amanda in her journey with our program. And thank you so much.
Hey, thank you. It was fun. Appreciate it.